Today we're going to look at how different types of cell integration will act on the perception or the overall distribution of action potential distribution throughout the network. So we call that neuron integration, and we're going to start by just looking at and reminding you about the types of chemical synapses. So we have axodendritic synapses, we have axiosomatic synapses, so an axon on the cell body, an axon on the dendrite, and here we also have an axon on an axon. So we have axiodendritic synapses, axiosomatic synapses, axioaxonic synapses here. We also have dendrodendritic and dendrosomatic, which are not demonstrated in the drawing, but they're also there. So these are chemical synapses, and I'll just remind you that we also have the gap junctions, or the electrical synapses. We haven't really looked into the effects that they have on neurotransmission just yet, but they will be covered in later lessons. Now let's start by looking at different types or different ways in which the flow of information through a neuron network can be influenced. And we'll start with different types of modulation that can happen at the synapses. So the first type that we're going to talk about is a presynaptic inhibition event. So here we have the presynaptic cell. It is going to fire an action potential or a series of action potentials down the axon. Normally it would create a response in the postsynaptic cell, something like this red line here. Okay. Now this yellow line is indicating the flow of calcium that's going into that synaptic area here. Okay, so in a normal situation without this other inhibitory neuron, so we have A exciting B, the excitation happens following the, so here comes the action potential. Once the action potential hits the postsynaptic cell, we see the postsynaptic potential will depolarize to a certain amount and then settle back down to the resting membrane potential. But as the action potential hits the synaptic area, we see this increased flow of calcium. So remember, the current is flowing in, so it's going down. Okay, so an influx of calcium that looks kind of like this pattern here. Now, once we put in this presynaptic inhibition, so now another neuron is going to inhibit the activity at this synapse in some way, usually with some kind of influence on the flow of calcium that's going into the synaptic area, which then would limit or reduce the amount of neurotransmitter release, because remember, the calcium influx at the synapse determines the binding of the vesicles and the release of the neurotransmitter. So if we have less calcium influx, we'll have less binding of the vesicles and less neurotransmitter release. So let's see what happens in the case of a presynaptic inhibition at the synapse. So we see basically at the postsynaptic membrane, there is the amount that was produced before. So that's the amount of depolarization. Now that amount of depolarization has reduced. So we see that that inhibition has reduced the amount of postsynaptic excitation. And if we look at the calcium flow, we can see that in the first instance where we had no inhibition, we had this massive calcium flow, this nice calcium flow here. Now we have a reduction in calcium flow. Okay, so that's an indication or an example of how we can have presynaptic modulation, and in this case, presynaptic inhibition. Now let's look at a different type of presynaptic modulation. This will be presynaptic facilitation. So in this case, we have our normal situation, A and B, and we see that when A excites, it causes a change in B of this much depolarization and this much calcium flow. Now we add in RC2, which in this case is a facilitator. So it's going to do something, it's going to release a neurotransmitter or a neuromodulator down here that's going to change the way in which the calcium current flows. It'll either increase it or prolong it in some way. Now in our example, we see that once the presynaptic facilitation happens, postsynaptically, there was our normal. So that was our normal amount of depolarization. And now with the facilitation, we see that depolarization increases significantly. And this is now a facilitated situation. But we look at our calcium flow. Previously, our calcium did this. Now we see that instead of returning to normal quite quickly, it's now stretched out or prolongated the amount of time it's taken for that calcium to return to normal. And during that time, more and more transmitter is released. And that causes the facilitation or the increase in the depolarization of the postsynaptic cell. So that's another example of presynaptic modulation, but in this case, it's presynaptic facilitation. Now let's look at how can we fine-tune responses a little bit in the nervous system. Well, we can use, depending on where the inhibition occurs, or the modulation occurs in the presynaptic neuron, we can see different global effects on that neuron. So let's take our example number one. So we have excitation coming down, it excites this neuron, and this neuron fires an axis potential. Now it has 3 collateral axons, okay? And those 3 collateral axons are going to 3 different synaptic connection areas. And you'll see that each of those synapses is going to affect a different effector. And in the case of the inhibitory neuron only inhibiting one of those collaterals, we see that in this case we have no response at one of the effectors, but the 2 other collaterals that did not have inhibition all produce a response. So this is a way of selectively inhibiting one collateral of this neuron. Now let's look at what happens if we inhibit the whole neuron and not just one collateral. So this is a different approach to inhibition, a different type of inhibition. So now we're going to inhibit the whole neuron. So here comes the excitatory action potential down. It hits this neuron, but because we now have an inhibitory action happening on this cell body, when it hits the axon production or the action potential production area, the axon hillock, it does not produce threshold stimulus. So we get no firing. Therefore, at all of the collaterals, we get no response. So here you see inhibition of the cell body back here can result in no activity here, whereas inhibition at one of the collaterals of that cell can result in a different array of stimulus or response at the other end. So again, we can see how inhibition at different parts of the network can result in grossly different response patterns. Now let's take, we talked about dopamine as a neurotransmitter, and we said it was a a very complicated and a very widespread and useful neurotransmitter. But for the most part, it modulated events at synapses. So let's look at, we'll take synaptic modulation, and we'll use dopamine as an example because it can do a variety of different synaptic modulations. And so we'll just look at some of the ones that dopamine can do. So let's take a look at the presynaptic terminal here. Now remember, in the presynaptic terminal, that's where the calcium will move in and cause binding of the vesicles and release of the neurotransmitter. Postsynaptically, we have the receptor sites, and so dopamine can modulate both presynaptically and postsynaptically. Okay, so let's look at how dopamine can do this. So the first thing that can happen is that dopamine can, by binding on this dopamine receptor, it can modulate the axon terminal excitability. So it modulates the flow of calcium into the synaptic area. It can also modulate the vesicular release machinery itself. So it modulates those SNAP proteins and those other synaptic membrane binding proteins that we talked about in previous lessons. So it can slow down or reduce the amount of neurotransmitter released by stopping the vesicular or slowing down the vesicular binding. Now, we can also have recruitment of postsynaptic dopamine receptors and liberation of retrograde signaling molecules. So the dopamine could bind sort of in this area here, and it can change the response downstream of the signaling molecules stimulus on the postsynaptic membrane. And there's a lot of different ways in which it can do this. It can modulate the sodium, potassium, or calcium channels in this postsynaptic area, not necessarily at the synapse itself, but in the actual postsynaptic neuron. So we have a variety of different things that dopamine can do, and you can see that dopamine, we talked about a number of dopamine receptors, D1 through D6, and some of these different receptors will do different activities depending upon how that receptor is wired to respond to the dopamine. So we can change postsynaptic activity with dopamine. We can change the activity in the actual postsynaptic neuron, not just at the synapse, but also in the neuron itself. And we can have presynaptic alterations by affecting the binding of the vesicles, by affecting the excitability level inside the synapse by the calcium movement, and also modulating some of the membrane potential situation. So by inhibiting or allowing the potassium to flow out at a greater rate, the dopamine can hyperpolarize the cell or inhibit the synapse so that it has to have a greater calcium input to cause vesicular binding. So there's a lot of different ways in which dopamine can act as a modulator. So this is just an introduction to how we can use modulation from dopamine to affect receptors in ways that you are now going to start understanding a little bit better, because now you're understanding vesicular binding, you're understanding the calcium flow at the synapse, you're understanding how neurotransmitters bind to receptors postsynaptically. So it's a very complex process, but we're starting to take it piece at a time and put the pieces of the puzzle together so that you can have a better understanding when we start talking about neuron systems in general and how they work. So let's look at a different type of modulation. This is called basically a recurrent inhibition. So neurons can feed back on themselves in a negative fashion. So basically this motor neuron will fire an action potential, and this axon here is going down on its way to a neuromuscular synapse or neuromuscular junction. But on the way down, this synapse develops this axon develops a collateral axon, which also excites this inhibitory interneuron, which causes feedback to go to the motor neuron that produced it, as well as motor neurons in the vicinity of the motor neuron that produced it. Now, this specific type of recurrent inhibition is a special example of a type of cell called a Renshaw cell, and it creates feedback and surround inhibition for motor neurons firing in the spinal cord. So let's just give you an example of how different types of inhibition can result in different types of activity at the end organ. Now, the next type of intracellular modulation we're going to use is feed-forward inhibition. So we had feedback inhibition on the cell that produced the activity. Now we're going to have feed-forward inhibition. So this is now going to affect a different cell down the chain that will change the activity in an effector organ based on the activation of a different neuron. So afferent, this is a motor neuron. Let's say this is a innervating neuron to a motor neuron. So this is going to be one of the integration neurons coming from somewhere in the spinal cord, somewhere in the brainstem, potentially from the cerebellum, and it's going to influence this motor neuron. So here's the motor neuron firing to the muscle, and in this case, this integration neuron will fire and excite the motor neuron, which is going to cause a muscle contraction. But this integrator neuron has a collateral, which also excites an inhibitory interneuron, which then inhibits the motor neuron of a muscle that is the antagonist of the muscle that was excited here. So sometimes we don't want our extensors and our flexors firing at the same time. Sometimes we do, but sometimes we don't. So in this instance, this system of feed-forward inhibition can create the situation where when an extensor fires, the flexor of that same joint is inhibited. So the muscles subtending that same joint are inhibited. And that results in an extensor result, or an extensor activation. And we will, for instance, push someone away or extend our legs or do something without the activation of the flexors causing some kind of interference with that activity. So feed-forward inhibition, very effective type of inhibition, used a lot in the motor system. Now let's talk now about disinhibition. Now this can be a very confusing concept when you first start, but let's take you through it one step at a time. So it's pretty simple when we have a feedback inhibition situation. So an excitation of an inhibitor, so an excitation of an inhibition neuron results in inhibition happening at some point down the chain. Now it gets a bit more complicated when we start doing inhibition of inhibition. So now we have an excitation of an inhibitor, which is normally inhibiting an inhibitor. which normally inhibits the cell again. So what happens now is we have excitation of this inhibitor, which now inhibits this inhibitor. So that results in less inhibition at this cell, or disinhibition. Okay, so if we have a constant firing rate and we inhibit the inhibitor of that cell, that firing rate will go up. Okay, so this is disinhibition. This is sort of used in the basal ganglia, which I will explain an example of next. But I just want to work through other types of disinhibition. So we can have disinhibition when we have two inhibitory neurons in a row, or we can have disinhibition when we have a single inhibitory interneuron which then inhibits itself. So feedback inhibition on itself, which results in, again, disinhibition at this end organ or end neuron. Now, we can also have feedback, so we can have inhibition of inhibition of inhibition. So this is now, I'll take you through what happens now. So we have an excitation of an inhibitor, which now causes inhibition of a cell that was normally going to inhibit another inhibitor. So at this point, we have an inhibited inhibitor. So at this point, the activity here increases in this neuron, which causes an increased inhibition at the endpoint. So an easy way or a simple way to remember this is just like you learned with integers in mathematics. When you have a plus and a negative, it becomes negative. So this is inhibition. When you have a negative to a negative, it becomes a positive. So addition of a negative becomes negative, addition of two negatives becomes positive, addition of two negatives becomes positive, addition of three negatives becomes negative. Okay, so that's kind of a quick way to get your head around what's happening with these strings of inhibition. And you'll see there are lots of networks that have complex inhibitory patterns, and that's because without inhibition in our system, without inhibition in our brain, things would spiral out of control into this huge, massive over-facilitation, over-excitation until the system couldn't handle it anymore. So inhibition is quite important in how our nervous system functions. So that's why we have different systems in place that have developed to help us have control over those so we don't spiral out massively into this big ball of excitation. So I mentioned I was going to show you an example of where we use these inhibition type things. And one of the most studied areas where we use this is in the basal ganglia. So in the basal ganglia, it's basically a very important deep nuclear structure inside the brain. That involves many different deep nuclei, in fact. And one of the major things that the basal ganglia does is important in putting things in the right order. So muscular movements in the right order, emotional content in the right order, even potentially memories in the right order. So the motor, so in this case, we're just going to talk about the motor function of the basal ganglia. And of course, basal ganglionic pathways have been implicated in Parkinson's and Parkinsonian syndromes. So movement disorders are quite common from the dysfunction of the basal ganglionic pathways. But I'll just take you through sort of the normal flow of how this pathway works. And you can see how the inhibition of inhibition actually adds up to result in an inhibitory pathway and an excitatory pathway. Now, let me just introduce the anatomy a little bit first. So we have the motor cortex, which is the area in the frontal cortex, just that where most of the motor pyramidal cells exist, that are going to be the excitation pathway to the motor neurons in the spinal cord. And they excite the caudate and the putamen, which are other deep nuclei. And that excitation of the caudate and the putamen results in inhibition of the globus pallidus internus and inhibition of the globus pallidus externus. So globus pallidus has an internal and an external component. The globus pallidus internus then in turn inhibits the thalamus. Now, normally the thalamus would excite the motor cortex. So in this case, we have the motor cortex exciting the caudate, inhibiting the globus pallidus internus, pars internus, and which inhibits the thalamus, which then causes a reduction in excitation in the cortex. Okay, now if we follow the different pathway, the motor cortex excites the caudate, the putamen. It inhibits the globus pallidus pars externus, which then inhibits globus pallidus pars internus. Now that means inhibition of inhibition. That means we have excitation of the globus pallidus pars internus, which means we have more inhibition on the thalamus, less activity in the motor cortex. So that's one pathway where we have, in the first pathway, we have inhibition of inhibition, which causes the thalamus to, so inhibition of inhibition causes the thalamus to release, causing increased activity in the motor cortex. Second pathway, we have inhibition of inhibition of inhibition, which results in an inhibitory overall effect on the thalamus, which less excitation in the motor cortex. Now let's look at, so the motor cortex also projects to the subthalamic nuclei, which then projects back to the globus pallidus and excites it and inhibits the thalamus. So we start to see how complicated these projection systems can become, and I'm not going to go through in much more detail. I just wanted to use this as an example of how we can use inhibition of inhibition to get basically a release of activity, or inhibition of inhibition of inhibition to get an inhibited activity, or when we simply have inhibition coming from the globus pallidus subthalamic nucleus, that's simply inhibition. So let's remember this in the future, because we're going to learn this in much more detail when we start to study the basal ganglia and its effect on how the brain processes information and the output that that thalamus gating has on the motor cortex and other areas of the cortex also, including emotional understanding, including putting memories in the right order, putting emotions in the right order, putting other types of functional things in the right order.